Welcome to chapter 12. 12 comes after 11, by the way. So we have chapter 12 is dealing with drugs. There's a lot of drugs that we talk about in chapter 12, and I'm not reviewing every single name of every drug in here. I am talking about the examples of them. You might review some of the other ones to be familiar with the names of them and where they might fall into these. Create your own table to divide up the names and which ones they might fall under, whether they're going to affect a cell wall, whether they're going to affect protein manufacture, or ribosomes basically is how that works, or folic acid synthesis, or cell membrane, being able to function, and then cell wall production. I already said cell wall, didn't I? Or nucleic acids, like DNA and RNA. I would create a table, and I might even make a table to fill out, if I was studying this for myself, that included the antivirals and the antifungals and the antiprotozoals and the anti-helminth drugs, just so that I don't get confused if I see them as choices on the test, and be familiar with them all at once. And then you can test yourself that way by putting your drugs. When I say make a table, you can even I would make flashcards. And then I would create I have a whiteboard. That's what I'm reaching for. So just a second. I'm not going to draw on it because I don't know where my markers are, of course, but just to kind of point out how I would do it, I might write out, why does it look like I'm looking off into the ether or something? Okay. So I might write down, the different, ways that antibiotics can affect cells. So I would have a category for cell wall, a category for cell membrane, a category for protein / ribosome, and whatever. I just read them all off to you. And then go through and try to put them in there. Now go through with your notes and go do it first, then erase it and see if you can take a picture if you need to, and then go through and see if you can do it again. Or if you want to do flashcards, just make flashcards with each of those topics on it and put them out on the floor and lay out the names of the antibiotics and see if you can do that. It sounds dumb and it sounds well, I'll just write it down on paper. That's fine if that's enough for you. A lot of times, and I'm not kidding, a lot of times people learn better when they do a little bit of activity with their learning. I'm just saying, you're adults, not your mother. I just suggest. So, it's never going to get back in there. The big concept here, when we're trying to talk about what antibiotic to use to treat, or what antimicrobial to use to treat any kind of infection, selective toxicity. It's a basic concept. It just means we want to kill the bad guy and keep us and everything about us safe. So maximal toxicity, that's the toxicity part. And it's against only the pathogen, hopefully. And that's the selective part. We're selecting to be toxic against the pathogen, minimal damage to the host. Remember that includes, we want to protect our own microbiome, whether it's our gut or our skin or whatever it is. Any of you ladies out there, probably you men, know a little bit about this, if you've known a lady. If you've ever had a yeast infection, a lot of times if you get a UTI and you take an antibiotic, or it could be an antibiotic for another infection, you take an antibiotic, it can kill out a lot of the normal healthy bacteria in, your genital area, your lady parts. And then when that happens, yeast, which is normal in your biome, that's not a bad thing. The yeast can then overgrow and then take over where it shouldn't have been growing that much. And that's where yeast infections come from. We get the irritation and vaginitis associated with that. Vaginitis, itis means inflammation. Anytime you hear itis on anything, inflammation of whatever the first part was. So vaginitis, inflammation of the vagina. So think about it that way. We don't want to kill off all those good bacteria that could be in, the genital tract or whatever it is. We want to keep everything nice and happy. This is how C. diff becomes a problem as well. We kill off the C. diff. We kill off the normal bacteria in the gut. C. diff has endospores. It's Clostridium. So remember, Bacillus and Clostridium have endospores. Clostridium difficile, C. diff, has endospores. We kill everything off in the gut. the C. diff can overgrow and overtake, and then that would lead to that pseudomembranous colitis. That's what it's called whenever you have C. diff overgrowth. It's the same concept as the yeast infection. Okay, so when that happens, that whole process is called a superinfection, whether it's with the C. diff or the yeast, whatever it could be. Okay. So don't forget about that. When we say we want to cause minimal damage to the host, we're also talking about the host microbiome. All right, we talked about the therapeutic index. This is the ratio of the dose of the drug that it would be toxic to humans versus its minimum effective dose. So if the minimum dose that you can take to get rid of the pathogen, the minimum one, is very close already to the dose that could cause toxicity in people, so bad side effects, then you're talking about something dangerous here. And you wouldn't want to choose that drug if you could help it, right? That's the therapeutic index. You definitely want to consider that as well when you are choosing your medications for your patients, especially if you have patients who are already weak for whatever reason, elderly, babies, women who are pregnant, their body is weakened because they're growing a baby inside of them, not to mention the baby inside of them could be weakened and could be affected by the antibiotics you're giving to the mother. So there's a lot going on there, and that is one of the things you also have to think about. Remember that antibiotics treat bacterial infections. That's it. They don't treat viral infections. They don't treat fungal infections. They don't treat protozoal infections. Okay, only bacterial. You don't give somebody who has the flu an antibiotic. If your doctor is giving you giving you the antibiotics for the flu, it's probably because he suspects you have a secondary infection with some sort of bacteria. A lot of times that happens, right? You get sick with the flu, and another bacteria that could already be in your body or could be introduced from outside can cause pneumonia. As a result, it can overgrow because your body's you're weakened, your immune system's weakened. It's an opportunistic thing potentially, and it could start overgrowing, and that can lead to development of pneumonia. That will be the doctor treating that, not the flu. So I just want to be clear. Okay. Common drugs to know for the antibiotics. Remember, we can treat production of the cell wall. We have treatment of protein production, folic acid synthesis pathway, cell membrane structure, and nucleic acids. So these are the 5 that can be affected by antibiotics, the 5 categories that we're talking about that can be affected by antibiotics, okay? Treating bacterial infections. When we talked about the previous slide, well, not to you guys, you probably didn't see it, but it was this microbial control one, and I'm just going through them. But in this one, we didn't really bring it up directly in this, but a lot of the chemicals and physical and mechanical means of controlling microbes, we divided them into the same exact categories, except for folic acid. Folic acid is a new one to the antibiotic category, okay? So that's the only one that's a little different. The other 4 that we talked about those. All right, the cell wall, the penicillins, and other beta-lactams. Remember beta-lactam ring, it looks like a square, and it looks like it's attached to a little house. I mean, that is what it looks like. So these are beta-lactam antibiotics. It's not just penicillins. There are other ones that fall into this category as well. But penicillins are probably the big ones to be aware of, okay? Then we have the ones that affect the ribosome. If it ends in cillin, it's a penicillin, by the way. Other ones we had talked about that affect the cell wall, or we had mentioned briefly that affect the cell wall that are not beta-lactam, don't have beta-lactam rings in them, might be like vancomycin or bacitracin. So we'd shown the structure of those in the slides, but well, yeah, be aware of them. The penicillins definitely know. You should definitely leave this course knowing that penicillins affect bacterial cell walls. If you don't, you're not trying. Like, this is a basic one. Okay, that's what I'm trying to say. Ribosomes. we talk about ribosomes because we're talking about protein synthesis, and most of the drugs that affect protein synthesis are going to inhibit ribosomal function in some way. Aminoglycosides, and then we had tetracyclines, glycylcyclines, those sorts of drugs, and some of them end in mycins, like erythromycin. These all fall under that category. Folic acid synthesis inhibition. These drugs are the sulfa drugs. They inhibit folic acid synthesis by sort of competitive inhibition. If you remember from a long time ago, when we talked about enzymes in chapter 8, competitive inhibition means that they're going to bind in the same site that one of the precursors for folic acid would, and therefore inhibit the pathway from continuing on. That's how the sulfa drugs act for the most part. This includes trimethoprim, sulfamethoxazole. Okay. Trimethoprim is just like the trade name, so to speak. Then we have the ones that affect the cell membranes. There weren't a lot of them that we talked about. I'd be aware of the polymyxins just because that's one of the ones that's in neosporins. We had the bacitracin from the cell wall category, and then we have the polymyxins here. And then we have the nucleic acid inhibitors, and these are going to include all of those fluoroquinolones. All right, so that's the antibiotics. The fluoroquinolones, by the way, they sort of mimic normal nucleotides, the A, T, C, and G. They mimic those, they look like that. They get stuck onto the synthesizing strand instead of the proper A, T, C, or G nucleotides. And then that makes it impossible for the cell to keep adding on nucleotides appropriately. And then that stalls DNA synthesis in the bacteria. Okay, moving on to antivirals. Antivirals are tricky because viruses hijack our own cellular machinery in order to reproduce. So if we're going to try to inhibit their reproduction, that's going to potentially cause problems in our cells as well, right? Makes sense. Antivirals that I want you to be familiar with. Tamiflu. Tamiflu blocks neuraminidase function in influenza. We will talk about this more when we get into talking about influenza in the final unit, unit 4. So this the last quarter of everything you're going to learn. But basically, influenza virus has this little bit on its outside, like a spike on its outside, that is called neuraminidase, and it helps clip it free. It stays stuck to the cells after it makes new viruses and buds out. It'll stay stuck unless neuraminidase clips it free. Neuraminidase has a bunch of other functions for influenza virus, but that's like the big one that Tamiflu really helps prevent. So it keeps them stuck to the surface so they can't go and infect new cells. Acyclovir. Acyclovir is an antiviral that blocks nucleic acid synthesis in HSV. That's herpes simplex virus. And not just herpes simplex virus either. It can be beneficial to be used in any herpes or herpes-related virus. That includes chickenpox, which is varicella zoster, which is a herpes virus, for example. There's other ones too, but that's just one example. Now we had the antifungals. A lot of those are going to end in azoles. Not all of them do, because, okay, so we have the azoles like ketoconazole. That's a pretty commonly used one. So ketoconazole is an antifungal. That's just one of them. Amphotericin B is not an azole, but you might want to be aware of that one too. That's a commonly used one. So those two are probably the bigger ones, ketoconazole and amphotericin. Miconazole, I think it's MY. I can't remember how to spell it, but that's basically going to be monostat for yeast infections. Okay, remember yeast is a fungus. Antiprotozoa, I'm just trying to give examples that you might have heard of. Antiprotozoal medications, quinine and other derivatives that end in quin, like chloroquine. These are medications that treat malaria. They can have pretty severe side effects like blindness. And then we had metronidazole. Man, metronidazole is a pretty useful one. It's great as an antiprotozoal medication, and it's also great for treating anaerobic bacterial infections. So it's kind of a more broad effective type of medication. Then we had the anti-helminth drugs. We didn't really talk about any of these specifically, but some of the ones that are used in veterinary medicine quite a lot and are actually used in human medicine quite a lot if we ever saw any, helminth infections in the United States, which you really won't be seeing any of that. But if you were to travel out of the country to some of the underdeveloped nations, they might need to have these medications to treat helminth infections, right? So worms. So this includes praziquantel and ivermectin containing drugs. The Kirby-Bauer assay, we use this in the lab. It was like a 3-day experiment where we spread bacteria out in a lawn on the, what is it called? The Mueller-Henson agar. There it is. I remember microbiology. We grow our bacteria out in a lawn on the Mueller-Henson agar. We stamped it with those little discs that contain antibiotics, and then the antibiotics basically diffuse out into the agar. And then based on how well that works at inhibiting the growth of the bacteria, you can determine how effective that antibiotic is, right? So that was the idea with the Kirby-Bauer assay, and that's what that's about. A side effect of these medications, the drug itself can be harmful. Its effects on the host body could be the harmful effect, like causing ribosome inhibition in the host whenever you're trying to only affect the pathogen, or people can develop allergies to the drug. It can also affect like the microbiome and cause problems with that. And we already talked about the superinfection in the beginning, so we're pretty much done with that. So that's that for chapter 12.